So plasma, the liquid portion. When we talk about plasma, when you hear the term serum, all right, serum says that that has absolutely no clotting factors in it. So when we talk about serum, basically it's what's left over, the remaining fluid, when blood clots, solids are removed. It's almost identical to plasma, but we have no fibrinogen. Do you guys remember him mentioning that? Now, do you think about tiny little fibers flying around in the blood in your vessels? You just don't think that sounds right. I don't, because to me, blood needs to move freely. And if you got fibers in it, how could it move freely? Does that make sense? Why do we need something, however, that could give rise to a fiber so that we can clot at the time of a breach into those vessels? Does that make sense? So, in this blood plasma, when it refers to serum, that's saying none of this clotting factor is present. Now, in plasma, we can say there are three major categories of the proteins. All right, doesn't mean that we've identified everything, okay? However, we've identified a lot, okay? And we've been able to say that for those proteins that we have in the blood, we have albumins, globulins, and the fibrinogen. Albumin, it's the smallest protein that will be present in the plasma, but it's the most abundant. Now, contributes to viscosity. Let's stop it right there. Viscous. How thick something may be. All right? Do you associate thick with the plasma? Yeah. So, I'm going to have to get you to think outside the box, because we are going to have to see what happens with this, with the viscous portion. Don't you love saying that? Everybody say it with me. Viscous. Viscous. You know, put some fun behind it. All right. Makes me think about the oil in my car. Just say it. Think about it. We're a car. We got to have oil, right? Just say it. Okay. Osmolarity. Osmosis. Think about what?
How many particles are in that solution?
Why does it make you think particles? Why would you think particles? What was an osmol? It was a substance that did not dissociate in the fluid. For example, if we have which one of those is going to dissociate? Well, osmoles wouldn't necessarily be something that doesn't, but which one of these will dissociate? Okay, raise your hand if you say NaCl. Raise your hand if you say C6H12O6. Wrong. This right here is my glucose molecule. Is that correct? This right here is my salt. Do we ever hear about a glucose molecule dissociating, breaking up? No. That glucose molecule will go into the cell, begin to go through the process of glycolysis in the liquid portion of the cell, and then if it needs to, move into the mitochondria to give us the ATP. My glucose does not dissociate or break apart until I get to the actual cellular respiration. So if I had glucose in a solution, I would have one osmole of the glucose. But have you heard of sodium chloride dissociation? Well, first of all, what does it mean to dissociate? Separate. Have you heard of sodium chloride separating? Why? What ions does it break down into? I got to have it break down into the sodium and the chlorine. For example, sodium, y'all didn't have me during part 1. Serena, you did. Okay? So you hopefully remember everything that we have talked about at this point has required the sodium. Yes, no, maybe? Yes. Not sodium chloride, but a sodium ion. So therefore, those two dissociate, and I would have 2 osmoles. Does that make sense? Now, what is the importance of maintaining osmolarity of my plasma? If this is my blood vessel, and this is my blood moving through it, I've already said viscosity, resistance to flow. Okay? Now, osmolarity will also be a part of that thickness. If something is in too high of a content, are we not going to mess up the osmolarity? We would therefore mess up the flow, which would therefore mess up delivery to the cell itself. Albumins are a protein, little tiny protein that's flying around in here, and their number can affect the flow. They're the most numerous, okay? Albumins, they are actually going to have an effect, because if we are thinking osmosis, okay, osmosis is our movement of water. The amount of water in the blood, important or not? Why? Stay hydrated and do what? So it can flow freely. Do you think about your blood flow? Start thinking about it now. All right? You need to stay hydrated. Because here's what happens. If you don't stay and hydrated means water. It does not mean soda, tea, coffee, these power drinks, everything else that's out there. Okay? It means water. If your blood is not flowing and it's not hydrated enough, it gets thick and becomes very difficult for the heart to pump, which means you're putting extra strain on the heart, and that's not good. Plus, you're not getting the flow to the tissue that you need. So, the importance of water, I can't state enough. But I'm once again, pure water, the water. Okay, not the water in a iced tea, not the water in a coffee, no. It's very important. Now, like I just said, having that effect will affect your blood pressure. The harder your heart has to beat, the more pressure we're going to have for the movement of blood. The flow, we need to keep the resistance low so that the blood flows freely through the vessels. And then fluid balance. If this is affecting the water level, we're affecting the water level on both sides of the vessels. So that little protein, it kind of is important. Our globulins. Globulins. Now, a globulin as a whole, okay, one of the things it can do, it can provide transportation. And when we look at this, okay, this is going to be transportation for something such as my lipids. Have you ever thought about transporting the lipids through the blood? The fats. When we break down our food parts and that gets absorbed into the digestive system, we have to transport those actual fat molecules to the area of the body they need to go. Most likely the fat cells. Okay? We got to transport the lipids. We have to transport carbohydrates. We have to transport hormones. We have to transport ions. We have to transport antibodies. So globulins, not just for antibodies, but globulins are for the transport of a lot of other materials. In the job for antibodies, the immune system, okay, different globulins are going to be present because antibodies, their structure, their function, their availability gets to be very specific. If the body has to produce antibodies, you're sick. All right? If we have to move past something other than a good old white blood cell, okay, chances are there's something very specific that has invaded your body, and the body has to mount the resistance against it, and it's very specific, which we'll get more into in the, I think it's not the next chapter, but the next one. Fibrinogen. Remember those little stem cells I mentioned? Okay. Fibrinogen, we need to make sure that it's available, because any time there is a break in any of these vessels, okay, we need to close it up. If we don't close up that vessel, we stand the chance of bleeding out. Not good, okay? Now you think, well, wait a minute, something such as it wasn't a paper cut, it was a cardboard cut. You know, paper cuts to me are like the worst, okay? But I was opening a package and I got the cardboard cut. And whenever something happens like that, oh, there it is, I couldn't see, okay? I needed my glasses. I got the cardboard cut. And see, you know, now that I kind of know what happens when we get cut or whatever, it's really kind of cool, because, you know, I'm sitting there and I'm looking at it and I'm going, oh, well, this is happening, how long should it take? Is it taking longer than it should? Blah, 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 blah. So, you know, it's kind of cool, you know? I look at it and I think all this stuff that goes with it. Now, we need those fibers present so that that process can start occurring. But someone who may lack this ability, or any other of the clotting factors that your textbook will get into great detail about, but I won't, okay? If they're lacking in those, that little cut right there could lead to them doing what? Bleeding out. Having to go to the hospital, to the emergency room, and getting the clotting factors. So it's a very interesting structure that we have here. These proteins that we have in the plasma, they're formed by the liver. Now, globulins, however, they're going to come from something called a plasma cell. That gets really fun. But albumins, fibrinogen are from the liver. Globulins are going to be from these plasma cells. So it gets really interesting. In this blood plasma,